Hi there, and welcome back to the Dutch Podcast. I'm Dr. Jacqueline Smeaton, and on this podcast, we dive into the vast and ever-evolving world of hormones as part of our lifelong pursuit of better health. Here at Dutch, we've dedicated over a decade to transforming lives by addressing hormonal imbalances like cycle irregularities, perimenopause, menopause, and more. Our mission is clear: to simplify and advance hormonal health for everyone. On this podcast, I get to bring you cutting-edge insights from our top experts on hormone therapy, menopause, personalized medicine, and more. We're all aimed at making a real difference in your well-being. Your questions and feedback are crucial. They shape our discussions and help us navigate the complexities of hormone health together. Now, as always, please remember, the content of this podcast is for educational and informational purposes only, should not be considered medical advice. Be sure to consult your healthcare provider for medical advice, diagnosis, and treatment. I'm so glad that you're joining us for today's episode. It's such an exciting topic and something that is really changing every single day with how we understand it. Today, we're going to be talking about the microbiome. If you've read the news or watched TV or followed natural medicine, functional medicine, you know the microbiome is everywhere. And today on the podcast, we have one of the top experts joining us to talk all about the microbiome and the relationship of the microbiome to things like stress, chronic stress, diet, and even things like weight loss drugs and the role that they might have on the microbiome. We get into covering things that I didn't know, and I've been learning about the microbiome for a long, long time. So I'm really, really excited to have our guest on today. I think you're going to walk out of here feeling like you've really amped up your expertise on the microbiome and definitely learning things that are both clinical and kind of the scientific background of why the microbiome is so important and some really specific examples of just how far-reaching the impact can be. Our guest today, Kiran Krishnan, is a research microbiologist and a health and wellness expert who makes complex information really understandable. He's founded a number of successful health and supplement companies over the last 20 years, including co-founding and leading Microbiome Labs, the preeminent microbiome therapeutics-focused brand among healthcare professionals. He's currently a co-founder and partner in 3 other companies that aim to revolutionize wellness care. He's conducted and published several research studies in scientific journals. He's published chapters in scientific textbooks and reference books. He has global patents and is a sought-after speaker on human health and the microbiome. We're really excited to have him here today. So welcome, Kiran. Good to have you.
It is great to be here. Thank you so much for having me.
So can you set us up by just talking a little bit about the microbiome in general? I mean, it's really it's been everywhere for the last 5 years,
Yeah.
maybe even more. And really, we've really come to understand the vital role of the gut microbiome in our life, in our health. Can you just give us an overview to give listeners a start?
Yeah, I think, one of the ways to really put it into context is to understand the scope in which the microbiome functions within the human system, right? So I always like to tell the story of our genetics because we intuitively have this idea that our genes dictate all our function. And that's true that our genes, of course, dictate function, but we have about 22,000 functional genes, which may sound like a lot if you don't know genetics, but an earthworm has 30,000 functional genes, right? So we're not that cool when it comes to our own genetics. We're not that sophisticated either. And in fact, this was a problem that came about from the Human Genome Project, where the idea was, let's sequence the entire human genome. We're going to find all the genes that do everything. And then, of course, for every disease state, there was likely going to be a genetic element, right? They're going to find the heart disease gene and the diabetes gene and so on. The estimate, I remember, because I was actually in school at the time, and one of the labs that was doing the Human Genome Project was a biochemistry lab that I worked in. The estimate was that we'd have over 250,000 genes minimum. And then they figured out that most of our gene is just kind of nonsense code that was spaces in between functional genes, and we had 22,000 genes. So then the big question was, how in the world do we do all the things we do? How are we so sophisticated if we're so lacking in genetic material? That was part of the impetus of kicking off the Human Microbiome Project, to figure out, do we have microbial genes in our system? And if we do we utilize them? And as it turns out, we have over 150 times more microbial DNA in our system than human DNA. We have over 2.5 million microbial genes. So when you look at the comparison, right, you have 22,000 functional human genes, you have over 2.5 million microbial genes, and those microbial genes dictate the vast majority of our functionality. So to be human, we require the microbiome, and we require so many elements from the microbiome, not only the genes that they represent, but then all of the byproducts of the fermentation and things that they create within the system. At any given time, if you pulled out, a liter of blood from your body and you examined all the molecules that are in there, around 50% of the molecules are there that are derived only from the microbes in the system. So we cannot overstate the importance of understanding the microbiome and understanding the role of the microbiome, both in our day-to-day function, in our health and wellness, and then, of course, in disease pathology as well.
So I actually didn't know that. That's really fascinating that we have more microbial DNA. I know we have more
Can you hear
microbial
me? No.
cells in our body than we
Yeah.
have human cells, which is one of the ways I talk about it with patients, but I had no idea. And now is that DNA, I don't want to take us off track too far, but this is fascinating. Is that DNA like an artifact of evolution, or that is DNA that is it integrated into our own
In our
DNA
own genome.
strands?
No, it's an artifact really of evolution. It's
Okay.
housed within the microbiome. So the word microbiome actually defines the microbes and their genetic elements. So that second part is
Mm,
really important there, as it relates to the host, right? So what you find microbial DNA and its consequences. So then RNA, microRNA, and so on that come about from the DNA. You find them, within our cells to a certain degree, not integrated necessarily within our chromosomes, but within our cells. You do also, of course, find them in the microbes themselves, and then the microbes do the translation to create the proteins that we need as a result of it. So there's so many functions that we require that we don't have genes for and we count on the microbes to provide us those genes. And this is why, the kind of thing that keeps me up at night is the extinction of the microbiome, right? Because when we lose components of our microbiome, we lose form and functionality, we lose capabilities, and that becomes really scary when you think about it. Because over the last, several decades, we've lost diversity in our microbiome quite a bit. And the analogy I always give to people is, imagine, based on our choices today, that our kids and their kids would be born without a spleen, or
right.
born with just one kidney, right? Or born with only half a liver. We go, whoa, what are we doing, right? We got to correct everything we're doing, because we can't have our future generations that significantly handicapped from day one. But we're already doing that by dismantling our microbiome and not passing on a really complete, healthy set of organisms to the next generation.
Yeah, well, it really reframes the like benefit cost impact of things like antibiotics and triclosan and hand sanitizers and all of the stuff that we've employed to kind of fight bacteria
That's
and
exactly right.
viruses, throughout the last few decades. It's fascinating.
Yeah.
Now, when we think about the microbiome, people always jump to the gut, but have there been, because there's a lot of research newer, has there been any organ systems where it's surprised you that they found a microbiome?
They've actually found a microbiome biota in virtually every part of the body. And when you start to understand the symbiogenesis, really is the fancy term of, how starting with our cells themselves, how our cells are really an example of an evolutionary adaptation of an archaea swallowing a bacteria, right? So hence the mitochondria within our cells are all ancient pleiotropic bacteria. So when you think about our system from a cellular level, and from that point on, we've incorporated microbes into everything, then it becomes not a surprise that you find microbes everywhere, right? In our brains, in our cerebral spinal fluid. There's actually a whole virome in your cerebral spinal fluid with lots of viruses that live there naturally. Your eyeballs have their own microbiota, right? Your heart, your lungs. The lung microbiota became a really big topic during COVID, because there were researchers that were starting to type what people's lungs looked like in terms of their microbiota, that were um, going into respiratory distress with the virus more readily than people that weren't, right? So virtually everywhere. And your skin is a complex area for microbiome. Your face has a very different microbiome than your arm, and thus it dictates the different types of tissue structures. And the thing that's super interesting to me about discovering that all different parts of your body, even in your GI tract, you have distinct microbiotas throughout the GI tract, right? The thing that's so fascinating to me is the biome of a given location dictates the function of that location. And if the biome changes, even though physiologically the organ itself has not changed, the function of the organ changes, right? And we see this all the time in the small intestine with SIBO, right? The small intestine has a very specific functionality, and it has certain things it doesn't do, and certain things it should be doing really well, like digesting food, assimilating nutrients, having a dynamic permeability within the lining, and so on. The moment the microbial ecosystem of the small bowel starts to change, meaning you get an overgrowth of the wrong organisms, the presence of wrong organisms, too much gram-negative bacteria, you get conditions like SIBO, which can lead to fatty liver disease and so on and so forth. So it completely changes how the small bowel works in the context of the microbiota. So everywhere in your body, we have a microbiota, and the microbiotas of those regions are critically important to how that part of your body functions.
It's totally mind blowing. I've really been diving into reproductive microbiome because I do a lot
Yes.
of reproductive medicine, and it's one of those areas where I was about to give a talk, and then like the week before my talk, there were like 3 new papers that I wanted to
Right.
incorporate. It's just so emerging, and
It is.
there's fascinating data on things like it's actually the microbial balance in the placenta that initiates labor.
Yep.
They're finding, and so if that's off, that can affect it. And they have found a microbiome in meconium, which
Yes.
is like the first bowel movement of a newborn, which
Right.
when I was in school, well, I was in college, maybe around the time you were, where like I was studying biochemistry. The Human Genome Project was published around when I graduated. I actually have it in Nature. I was a subscriber to Nature. I
Oh,
have
cool.
that journal
Yeah.
still wrapped in plastic because I'm like, this will be worth something someday,
Totally.
at least to nerds like me.
Yeah.
It's never been opened or never been touched. But my point is that it's just things are emerging so quickly. And, we used to be taught in naturopathic medical school, this is 15 years ago, that the uterus was sterile and you
Right.
treat it as sterile, and that babies really colonize their microbiome through the vaginal canal. And now we know that's definitely not true.
Uh-uh.
It's just, they do, but they also have one in utero. There's just so much
They do.
around this that we could talk about.
Well, and there's one more thing when the talking about the brain, right? So we'll talk about the gut brain axis at some point, but speaking of in utero, how important the gut brain axis is even there. So there's evidence now that mom's immune cells, so in particular dendritic cells, will go to the terminal end of her small bowel, the ileum, and reach across and grab certain types of bacteria, and it knows what type of bacteria to identify. And these are typically bacteria that produce a carbohydrate called peptidoglycan. So they're unique bacteria, gram-positive bacteria with a unique carbohydrate structure on the outside. It'll reach across, grab those bacteria, take it to the cord blood, and drop it off at the placenta. Now, what's fascinating about it is that dendritic cells, which are antigen-presenting cells, right, their job being to grab things that are potentially dangerous and then alert the immune system and present the antigen to T cells and B cells. That's what they do 99% of the time. But in this context, they don't alert the immune system at all. They grab these bacteria, they digest the bacteria on the inside of the dendritic cell. They go to the placenta, and then they squirt out this peptidoglycan, these carbohydrates from the bacteria. These peptidoglycans then have receptors on the placenta to grab them, and then there's transporters that take the peptidoglycan to the baby's brain, and the baby's brain has receptors for bacterial peptidoglycan, right? And when it binds the receptors, it turns on things like the formation of the corpus callosum, the formation of the blood-brain barrier, and differentiation of the different lobes of the brain and all that, right? So from day one, the development of the human brain in utero, which is maybe arguably one of the most important things we want to happen appropriately in utero, we've outsourced that, and we're dependent on bacteria for some of that stimuli, right? So that the connections are mind-boggling when you think
That is
about it.
mind-boggling. I mean, I want to talk about this all
Yeah.
day. How much time do we have today? No. No, but it is really fascinating because it's like an iceberg. I'm sure that 10 years from now, we're going to look back at today and say we knew nothing back then.
Totally. Yeah.
It's just really amazing to think about the impact and the symbiosis that happens. Yeah, of course, it makes sense. But to be able to uncover that and understand it better is exciting.
Yeah.
So you mentioned the gut brain axis. So I do want to talk about that today because I think that's so interesting. And I'd like to really start by just painting that picture. People have probably heard that, the gut brain axis, but what does that even mean?
Yeah, super important. So there's a number of physiological connections that become really important in this context, and then there's some biochemical components of it as well. So the physiological connection being that the digestive tract is covered with the second highest density of nerve endings, um, being the enteric nervous system. The enteric nervous system almost being thought of as a second brain. This massive high density nervous tissue that covers the entire digestive tract. That enteric nervous system is then connected through the vagus nerve directly to our brain. And there's, in many respects, we can look at parts of the central nervous system and the brain and the spinal cord and all that as and the enteric nervous system as kind of being two parts of the same thing, right? They're not actually two different units and all that. They're really two continuous parts of the same thing. And it's a bi-directional highway. So as much as information travels from the gut to the brain, so does information from the brain to the gut. Then, of course, because there's information going from the brain to the gut and the gut to the brain, you've got microbes that cover the gut in every square millimeter of the digestive tract, right? You've got something like 30 or 40 trillion bacteria there, and they have direct access to this enteric nervous system. And then what's another fascinating aspect of all of this discovery is within the lining of the intestines themselves, there are all of these enteroendocrine cells, right? So these are essentially neurological endocrine cells, meaning they have very specific nervous function and nerve ending function, as well as endocrine function, right? So they're super unique in that they can produce hormones and prohormones, but then they also produce neurotransmitters and conduct neurotransmitters and so on. That actually becomes important in the GLP part of the conversation. But what it shows, the fact that we have these types of cells in our gut lining, is that our gut is the major site of understanding what the world is like that we live in, right? Our gut is the biggest sampling site for our body. That's where we physically encounter most of the things out in the outside world through food and breathing stuff in and drinking things and all that. And those cells are constantly monitoring what's happening with the host and what the host is being exposed to. Those signals are being sent to the nervous system, being sent to the brain, being sent to the immune system. And there's a constant communication there to help the host adapt to the situation. So that gut-brain connection cannot be overstated in terms of its importance in maintaining function of the brain and thereby also maintaining function of the gut.
So talk a little bit about when we think about with the Dutch, one of the things we look
Mhm.
at is stress, right? We measure cortisol production. And can you share a little bit about the impact that stress has on the microbiome? And what do we
Yeah.
know and what do we suspect? Because I think there's probably a gap there as well.
Yeah, so in 2015, and this was actually for me, I started really, really working deeper into the microbiome probably in 2010, and lots of that information was very, very nuanced and new then, and then the sequencing technology wasn't that great, so
Hmm.
so it wasn't really great information, but there was a lot of suspicions at that point. But where the game really changed was in 2015 when there was a publication in a journal called The Frontiers of Immunology. And this was a meta-analysis paper at that time that had reviewed a number of papers that fit within this topic. And they concluded that stress-induced intestinal permeability was the number one cause of mortality and morbidity worldwide, right?
Wow.
So,
Unbelievable.
which is crazy.
Yeah.
Stress-induced leaky gut and they went through all the pathology and pathways and how all that disrupts the microbiome and causes disease. That was the number one killer worldwide because it is at the root cause of diabetes, heart disease, obesity, cancers, autoimmune conditions, you name it, right? They were able to connect the pathologies and pathway to all of those disease states. So stress is one of the most potent drivers of dysbiosis in the gut. And it's also one of the most potent drivers then of eventual leaky gut or intestinal permeability. So how does that happen, right? And stress is of course an issue of the balance between sympathetic and parasympathetic, overt expression of the HPA or activation of the HPA axis, and then of course you've got cortisol cycling and movement. But at the end of the day, from the microbial standpoint, and we'll talk about how microbes intervene in stress. This is where the role of psychobiotics have come in as well. That's a fascinating area. But overall, what tends to happen when you experience small bouts of stress throughout the day is that there are opportunistic organisms within your system that have been designed by nature to read the stress signals from the host as the opportunity to express their virulence factors, because they've come to learn that when the host is stressed, the host's immune system is compromised, right? And so we know this. People who have, herpes simplex, who get cold sores, when they're stressed, the cold sore pops out. People with Epstein-Barr, cytomegalovirus, right? These long-term latent viruses, when they're stressed, the infection comes out. When people are stressed, they get acne. You get stressed, you're going to get sick, right? You're going to get more susceptible to illness. So we know this to be true, that these opportunistic organisms express their virulence factors and the immune system is suppressed. So there was a few studies that have shown that little bouts of stress throughout the day is like taking little doses of antibiotics as you go along. Now, on top of that, our behaviors when we're stressed further damage the microbiome, because one of the things you're not doing when you're stressed is you're not sleeping, right? And we know that sleep, that period of rest, is really important to activate and proliferate certain microbes within the microbiome. We also know that when you're stressed, a lot of people eat and drink excessively or do things to try to stoke some sort of dopamine to reduce the stress. All of those things are damaging to the microbiome. And so stress is arguably one of the most potent drivers of dysbiosis, and then all of the functional dysfunctions that occur as a result of that.
That's so fascinating. I personally had that experience where I went through a stressful time and then lo and behold, got SIBO. And it's like, where did this come from? I'm like exercising, eating the same, everything's the same. And it was just that event. Like I've experienced that myself. It's just a fascinating thing.
Yeah.
That it can be so impactful.
Yeah. And we've always known it, right? Like, I mean, it's, your moms and grandmas and all will tell you also like, oh, manage your stress. And people kind of knew it intuitively that it's not necessarily a good thing for our physiology to be stressed all the time. But now we have like real biochemistry and microbiology answers as to why it's so bad. And we sometimes kind of take it for granted, and don't really pay attention to it the way we do maybe to diet and other things as well. So that stress management and figuring out how to deal with that, I think is one of the most critical things that people can do.
I agree. And I totally agree with you that even like clinicians, nutrition and movement get prioritized over stress management. And I think another piece of that is that we don't do a good job preparing clinicians to provide tools and resources and solutions.
Yeah.
We like our adaptogenic herbs, of course, but what are the behavioral things that patients can do to manage stress? And a lot of us aren't practicing them because we are out of med school, running businesses, all that stuff. So I absolutely think it's one of the hardest things to counsel patients on. But like you said, one of the most critical for overall health. So it's so interesting to learn more about the microbiome connection as well.
Yeah.
So what happens over time? Like this is what you've described as happening with short-term stressors. We see this kind of antibiotic effect where you can get this growth of pathogenic organisms. What about gut diversity and microbiome makeup over time if people aren't managing stress? Does that compound it and really change things?
It is, yeah, because in a way it's a self-perpetuating cycle, right? So what tends to happen is that when you experience bouts of stress, you get certain organisms that are favored in that environment. So they express their virulence factors, they start to take over more real estate. Now, those organisms also want the host to continue to be stressed, so they actually have mechanisms to create neurotransmitters and compounds that can actually drive more anxiety in the host, right? So they try to create a basal level of anxiety in the host by activating the sympathetic nervous system, by creating toxins and neurotransmitters that activate the HPA axis and so on. They also use inflammatory cytokines, for example, IL-6. When that's elevated, IL-6 will activate the HPA axis as if it's an external stressor doing it. So then you have this period where your little bouts of stress are creating a favorable growth environment for certain groups of bacteria and potentially viruses as well. And then those bacteria will do further things to maintain that stress state because that's a better environment for them to grow. And as you maintain that state, they take over more and more and more real estate. So it creates very significant dysbiosis over time, shortens diversity quite a bit. Keystone species like Akkermansia and Faecalibacteria, those types of microbes tend to suffer under that condition. So then they lose real estate and they lose a footing in the gut microbiome. And then eventually you start to see all of the pathways and pathologies that become a result of that, which is chronic low-grade inflammation, leaky gut, a dismantling of the mucosal structure, the lining of the gut. You start getting immune dysfunctions because now you don't have a proper microbiome communicating and working with the immune system. So the immune system's kind of going haywire and attacking everything it sees, right? So you may develop allergies, asthma, eczema, psoriasis, IBD, all kinds of conditions. So it's a very, very potent pathway and a very common pathway towards long-term dysbiosis in certainly in the modern world.
It seems like a hostile takeover.
It is, yeah, it is. And it's a
They come along
self-perpetuating cycle, right?
Yeah.
Because as those microbes take on more and more real estate, they are better at putting and keeping the host in a stress state because that's better for them, right? And then because you're in a stress state, it allows them to grow even further, and it just keeps going in circles.
Wow, crazy. So I want to now shift gears a little bit and talk a little bit about something that's really rising. Everyone's talking about, GLP-1 agonists like
Uh-huh.
Ozempic and the semaglutide, tirzepatide, all these things that are out there on the market. Has there been any data on the impact of those drugs on the microbiome?
So there's been some short-term data in people that are severely morbidly obese or very, far stages of diabetes, and they've been insulin dependent and so on. In those cases, they've shown some improvements in the microbiome in short term. However, there's very, very good reason to think that these could be really dismantling for the microbiome over time, especially in non-morbidly obese individuals and people who aren't, type 2 diabetics or have been type 2 diabetics for some time. And that's where I see the real danger in some of these things, right? Because we are utilizing a really powerful medication that really arrests very normal processes in the body, right? We could break down what those are in individuals that probably don't need it. Right. To me, it's the same as using insulin injections in a non-diabetic. Right. It becomes really a profound, change in how their physiology works. If you are an insulin dependent diabetic and you're no longer producing insulin, then you need injections. But if you're just someone who's like, has an A1C of like 5 or something like that, and you're producing insulin, but you have bad habits, and then you start injecting yourself with insulin, you're going to get some really negative side effects, right? So that's kind of where I see it. I think there's certainly people who need it, and there are people who will have an overall health benefit because they are morbidly obese and they are struggling, and all of the risk factors that obesity presents to them are probably outweighing the risk of the drug. But then what we're seeing today is that those aren't the majority of people that are using it. It's the people who want to lose 20 pounds. It's people who want to lose 15 pounds. And I personally have friends that I know that are in that category, that are in the I need to lose 20 pounds category. And a year later, they're still on it and they're like a fragment of what they used to be. And so I think there's a lot of issues.
Size, what meaning they've lost more weight or their health's declined
Yeah,
or
both,
mm,
right? So,
yeah,
because one of the problems with it, of course, is that you lose muscle
right.
almost equivalent to fat, right? It's like a 40-45% muscle to fat loss. And so when you take an individual who's like, for the most part, of normal body weight, yes, they might need to lose 15 pounds or so, and then they're on it for 4, 5, 6 months and beyond, and they've lost half of that weight as muscle, then they become like a fraction of themselves
Right.
because they become, and you lose but that's where the Ozempic face comes from, right? Because you lose muscle in your face and places that you really don't want to lose muscle, and you just
Mhm.
become so gaunt. And then the energy levels, and we'll talk about glucagon and the ability to actually store glycogen in the muscles and the liver and so on. And so they have very low energy. They have zero appetite, right? They used to be foodies actually to a certain degree and really enjoy going out socially and all that. So they don't have that anymore. And then it seems like there's a switch in risk of addictive behaviors
Mm.
as well, right? Because for individuals that were getting a lot of dopamine rewards from food, and if you all of a sudden cut that out and there's no reward sensation at all from food, then there is a risk that you start leaning towards other sources of that dopamine kick and that reward, right? This was seen in another drug that actually never ended up getting approved, and this was a drug that was an antagonist to the endocannabinoid system, right? So these GLP-1s are agonists to the GLP-1, so they stimulate the GLP-1 receptor. This drug was a antagonist to the endocannabinoid because we all know when you activate the endocannabinoid system, it increases hunger and it
Mhm.
increases your desire to eat. This is why cancer patients will smoke marijuana to try to improve their appetite, right? So the idea behind this drug was that let's take the appetite component out, right? Let's bind all the endocannabinoids and receptors and take out the joy of food, and people will not want to eat and they'll lose weight. And sure enough, people lost weight because they lost all desire to eat. But in that case, the reason the drug didn't get approved is I think it was almost 30% of the individuals started going into deep depressions and suicidal ideation and all kinds of stuff, right?
Mm.
Because you're screwing up like a really important aspect of reward centers of the brain and the central nervous system.
Yeah, it makes sense. So let's talk about the improvements in people who are morbidly obese with the
Mhm.
microbiome. It. Do you think it's an effect of the drug, or do you think it's an effect of behavior change, reduction in weight, reduction in inflammation? It's probably indirect. That's what you're implying.
That's right. Yeah, because the drug itself, by binding GLP-1 receptors, you're not doing anything directly into the microbiome. So it's a result of them reducing the caloric intake, reducing the types of foods that they're eating. They're reducing sugar intake, they're reducing calories just in general. And we know that when you overeat, and this is one of the effects of endotoxemia, we know that when you overeat, it causes dysbiosis. And the
Mm.
reason for that is a really high caloric intake causes a lot of disruption in the gut because it's a lot of food that sits there for a long period of time. There's a lot more hydrochloric acid being spilt out, a lot more pancreatic enzymes, a lot more bile salts. All of those things kill microbes as it's trying to break down and digest food. And then there's also an inflammatory response to the presence of that food, especially if they are foods that have really bad fats in them, right? Oxidized fats and so on. So you're going to induce an inflammatory response during that digestive process. That inflammatory response and the additional length of digestion causes dysbiosis over time and has been shown to be one of the mechanisms that drives leaky gut. And
Mm.
so they're changing those behaviors just because they're just eating less and their bowels aren't moving, they have no appetite. And I think that's where some of the improvements are coming from. But there's also indication now that is a diminishing return. But at some point you'll start to see a reverse, where whatever benefits you saw early on will probably propagate to less beneficial
Yeah,
over
that
time.
makes sense. And I also think about, if you're if you're morbidly obese and you're reducing your caloric intake, you're also reducing, most likely, exposure to ultra processed foods,
Yes.
which are also detrimental to the gut microbiome, just with the additives. And there's so much in there that's harmful. So that's
Absolutely.
probably another piece, is just like less exposure to harmful
Yeah.
things. So yeah, I can really see what you're talking about here, that there's a benefit initially, but if you stayed on it long or you weren't an appropriate candidate for the med, and they are used, I mean, I know my Instagram feed is like covered in ads,
Yes.
as is probably everybody's, trying to sell it to people who maybe really don't need it or aren't the designed patient for it. Um, and really could be causing more harm than good.
Yeah. Well, and and the thing is, like, it was developed as a diabetes drug, right? So the semaglutide was really developed as a diabetes drug, and the dosing as for it as a diabetes drug is actually much lower than the dosing that's given now for weight, and the dosing that people have access to just by being able to buy it online. And the other thing that really worries me about it is that, to me, anytime we circumvent a normal process of the body in a very dramatic fashion, we're absolutely going to cause a problem. And maybe we don't see the problem now. Maybe we can hypothesize what the problem will be. But I think history has dictated that we will absolutely create a problem, right? So there's two things that really concern me with this, especially long-term use in people who are not severely morbidly obese, who are like on death's doorstep because of their obesity. Um, the two things are, number one, so GLP receptors, when they bind natural GLP that we make and our microbiome makes all the time, these are they bind them for minutes, right? Maybe hours at best. The binding affinity of GLP agonists is really low naturally. So it's the receptors are designed to bind something, and then the thing releases,
Let it
right?
go.
And so the way the semaglutide and these other things work, and the reason why you can do once a week injections, is they bind for a week, right? They've been able to tweak the binding affinity of these peptides so well that they sit on the receptor for days, and that doesn't happen naturally, right? So what is that going to cause when we completely circumvent the normal biochemistry of how these receptors are supposed to work? So that's one big issue. Second big issue to me is it skips the vagus nerve,
Hmm.
right? Because the way it's supposed to work is all the satiety signals and the binding of GLP-1 receptors in the brainstem comes from the gut, moves up the vagus nerve. There are checkpoints along the way, and then it gets into the brainstem, binds it, and then sits there for a little bit, creating the satiety signals and the release of leptin and so on. But it's regulated from the gut, right? And then that is part of what creates the sympathovagal tone. That's the tonality of the vagus nerve. And of course, we know that the vagus nerve is a really important part of the autonomic nervous system. So now you've got the vagus nerve balancing between sympathetic and parasympathetic, and the tonality of the vagus nerve is so important for function of the digestive tract, function of mood, managing the gut-brain axis, and so on. So now you've got these peptides that skip that process and go right to the brain stem and bind and sit on those receptors for a while. There is this risk that you actually start to lose function and tonality in the vagus nerve. And we know that that is already seen in a certain percentage of cases because you start to get really slowed down bowel movements, right? You get chronic constipation, and the percentage of people end up with paralysis in their digestive
Wow.
tone, right? Which means that even 6 months after coming off the drug, their bowels are no longer moving, right? So they're going from gastroparesis to basic gastric paralysis. And that is a function of disrupting the tonality of the vagus nerve. And so I think, those are the kinds of things that, in my mind, when I look at something like this, I go, we're playing with fire, because we're circumventing things, we're doing things on receptors that are very unnatural. And anytime we've done those kinds of things in the past, we've created more problems than we've solved.
Yeah, it's like, just because you can do something doesn't mean you should, right? And we
Exactly.
see that with like you're making me think of like the Celebrex
Yeah.
Vioxx, where you like shut down this inflammation pathway, but you don't, and it seems so positive, but you don't realize all of the unintended consequences of that
Yep.
biologically. And I think if we can guarantee anything, it's that our biological systems are really complex
Hmm.
and hard to understand and hard to predict and foresee the impact. So I really am hearing what you're saying,
Yeah.
which is not that these are bad, which is just that they need to be used appropriately for patients
Exactly.
that are,
Yes.
that they're intended for, really.
Yeah.
Now, this is something, one thing that I've seen is like natural products that are marketed for GLP support. And I've seen probiotics as well. Are there species that tend to, I don't know, create fermentation byproducts that bind to GLP-1 receptor?
Yeah, there are. So there are, for example, one of the bacterial byproducts that is a GLP-1 agonist is indole. Indole is a compound that can be produced by certain classes of bacteria within the gut, Clostridia being one of them. E. coli produces indole. So those bugs can actually create indole, and then that can act as a GLP-1 agonist. There's a tight balance there, though, because you can
Yeah,
easily produce too much indole
right.
and it creates massive inflammation, all kinds of other problems. Short chain fatty acids also have, of course, the benefit of acting like GLP-1 agonists. So butyrate, in particular, can have that effect as well. And those are generally good for you anyway, short chain fatty acids. So some of the keystone species like Faecalibacter acromantia do that at a higher level, but you won't find them in probiotics because those are all strict anaerobic bacteria that cannot be manipulated outside of the body. They die very quickly when they're exposed to oxygen. So the likelihood that the mechanism of action is through short chain fatty acids is probably pretty low. The other class of bacteria that can actually produce a compound that act as a natural GLP-1 agonist is hydrogen sulfide. So you take sulfate-rich foods like mushrooms and things like that, and then there's sulfate-reducing bacteria that convert that to hydrogen sulfide, and hydrogen sulfide can act as a GLP-1 agonist. Again, that's a very tricky balance because too much hydrogen sulfide will create dismantling of the lining of the gut and all kinds of inflammation. So there's a balance there. The best I've seen in terms of natural products with GLP-1 effect is like a hops extract, a fermented
I've
hops
seen that.
extract, right?
Hmm.
Yeah, and they've got something like 8 published studies on it. What's interesting about it is it's not just a GLP-1 agonist, it also upregulates other receptors like PYY and a few GPR. So GPRs are G protein coupled receptors. So they have other components of metabolic health in that they seem to improve. And they don't have the same binding affinity, right? So they don't sit on the receptor for a week. It's a few hours, so it's a little bit more natural. And I think something like that can help. I tried it just to see, and I did feel on one dose, one tiny little capsule, I felt like my bowels weren't moving for
I
like
did too.
the
I've
rest
tried
of
it
the day,
too.
right?
I
Yeah,
got a sample, like a full size 30-day sample, and I tried it for like 4 or 5 days, and I'm like, I'm not hungry. And it
yeah,
was that effective. I could have just not eaten all
totally.
day. So maybe I would do it if I was trying to do a 24-hour fast or something like that.
Yeah,
But it was, I mean, I love food. I love cooking. I love food.
same here.
I like to eat food. So I like having a healthy appetite.
Yeah, and,
It's
and 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 then of course you have ample choices to make the healthier choices and so on. And I think that's a more meaningful struggle for me than doing something that circumvents my joy and
yeah.
desire for food, right? Because that to me was the most bothersome thing in seeing friends who aren't the people that need these peptides, right? They're not the morbidly obese people, the people that are on death's doorstep because of metabolic disease that could be rescued from these peptides. They are the people that want to lose 15 to 20 pounds. And it has taken all of the joy out of going out to dinner with them and, like just spending that social time around food and, drink and things like that. That has all just been completely attenuated,
Mhm.
which is a shame to see that.
Yeah. Fascinating. So, pulling us back to the microbiome, it really sounds like, and I'd love to hear your prediction. How will our increasing knowledge of the microbiome transform medicine? Like, what's going to be different in 10 years?
Yeah.
Because I imagine you probably think about this when you wake up in the morning and fall asleep at night. how is medicine going to be different? Are we embracing, is conventional medicine, conventional research embracing this connection? And what kind of impact do you think it's going to have on our healthcare system?
I think it will have a significant impact. I think two things should generally will change. Thing number one is that almost anything that we bring to market for human use and human consumption will need to be studied against the microbiome as part of basic safety, right? That's one of the problems we're facing right now in the world that we live in, is that the vast majority of things that we consume, we put on ourselves, we put around us and all that have never been studied on the microbiome. So we don't know what the effect is, right? Like, for example, glyphosate,
Mm.
Roundup, right? The whole premise around Roundup being safe is that it interferes with a biochemical pathway called the shikimate pathway that humans don't have. So the whole idea was that, hey, this interferes with insect biochemistry. It doesn't impact the human cell because we don't have this pathway, and that's the basis of its safety. However, most of the microbes in your gut have that pathway, and it dramatically affects them, right? So
Mm.
now we know that it can have a huge impact on the microbiome, and then thereby the host as a result of it. So I think it'll be things like that where we start to see that a basal level of safety analysis on compounds, on drugs, on food, and all that will have to go through a microbiome screen, right? As we come to understand the impact of the microbiome in health. So I think that's one aspect. The second aspect is medicine is starting to leverage the power of the microbiome more and more in order to effectuate certain pathways that we want results from. A good example of this is in cancer immunotherapy. I've had the pleasure of working with a couple of different cancer docs who are really pioneers in immunotherapy. And what's so fascinating about it is, like, there are a couple of cancers that can do really well with immunotherapy. Take melanoma, for example, a non-small cell lung cancer. But what was always confusing with this is, like, you took 100 patients and you gave them all immunotherapy. Somewhere around, you know, 30-40% of those patients will do amazingly well. The immunotherapy gets rid of the cancer, it never comes back, they have very little side effects, and so on, right? Then there's a percentage of them that has no real effect, and then there's a tiny percentage of them that has a toxigenic effect. And so the big question is always like, what is the difference between those that have effect and don't have an effect? What they're coming to find out more and more is because immunotherapy counts on our own T cells to function
Mm.
properly, is that the microbiome of the individuals is what determines to a degree how well the therapy works. So then in the world of cancer immunotherapy, they're going, well, we got to enhance the microbiome before doing the therapy so that we can get better results out of the immunotherapy, right? So that is a good example to me for the medical community starting to go, okay, part of the reason why our drugs work differently in different people is because of the microbiomes of different people. And if we can do some things to support or help or improve the microbiome, the drugs may work better. I saw this in a research project we were starting at University of Florida as well, right before the pandemic. So the pandemic kind of killed the project, unfortunately. But this was their top trauma surgeon at the University of Florida, and one of the things that he realized is that anytime he did trauma surgery, admitted the patients for post-op care, and then looking at the risk of complications, how long it takes them to recover, and what the rate of administration or administration into the hospital after they're left the hospital. All of those things are dependent on what that patient's microbiome looks like,
Mm.
right? The healthier their microbiome was, the lower their IL-6 levels and inflammatory cytokines were post-operatively, and they did better. They recovered faster, and their revisitation rates or readministration rates were really low. And so we were starting the study of, like, okay, before someone goes into trauma surgery or during or right after, let's enhance their microbiome with prebiotics, probiotics, to see if we can reduce all of those complications. So there's definitely interest, and there's definitely a line of sight to how medicine can improve significantly if we think about things in the context of the microbiome. And there's a good amount of work going on in this space that I think will materialize in the next 10 years.
It sounds like there's such an opportunity for personalization of medicine,
Yes.
really aligned with the microbiome. And, you know, one of the things that I find so fascinating is that there's differences in patterns of microbiome, like geographically,
Bring
based
it
on
in.
ethnicity, obviously dietary patterns. are we going to be able to get to a point where we can like type a microbiome, or where is that going when we talk about testing and like
Yeah.
what clinicians can do? It feels so complicated right now because it doesn't seem like there's a very clear target, right? We have some
Yeah.
target species we know that are keystone that we want to have enough of, but I think our clinicians would really love to know your perspective on that. Like, is there a goal? What are the key markers they should look for?
Yeah.
And will we get to a point where we can really understand what healthy looks like for a particular individual?
Yeah, I think we absolutely will. We did a lot of work when I was at Microbiome Labs on understanding functionality within the microbiome and trying to type and map out that functionality, right? So the difficulty about the microbiome is that most of us at a species level can be vastly different in terms of distribution and the types of species that we have. But what's really interesting about the microbiome is that functionality is conserved, right? Which means that we all have a bunch of functional redundancies within the microbiome. For example, you might have a whole different set of bacteria that make short chain fatty acids than I do. Let's say we both make, X number of grams of butyrate from X number of grams of fiber, but the types of bacteria and the proportions of the bacteria that do that for each of us can be completely different, right?
Mm.
And so the initial tendency was to go, okay, let's figure out at the species level what is a healthy microbiome. Okay, you have to have this species and that species and that species. Then we come to find out that everybody's got a different array of species, right? So let's then look more at functional groups of microbes. So then we started with the biome effects test that we had developed. We started creating patterns around functionality of the microbiome and incorporating all the bacteria within a certain group that did a function. So, as an example, you might have sulfate reducing bacteria that's made up of 9 different strains, and I might have sulfate reducing bacteria that's made up of 6 strains. And they might be very different strains, but the relative abundance of our sulfate reducing bacteria to the rest of the microbiome is the same, right? Which means your 9 species, let's say they make up 2% of the total microbiome. My 6 species make up 2% of my total microbiome, which means both our biomes have the same tendency to reduce sulfates in foods into hydrogen sulfide, right? Same thing with ammonia producing bacteria, vitamin producing, estrobolome, short chain fatty acids, all of the different functionalities that we count on from the microbiome. So I think where we'll get really good about this in the next few years is typing the functionality of your biome. And it may be done differently in each individual's biome, but the result of the functionality would be the same. The analogy I like to use is like if you had a Apple phone and a different brand phone, right? They function differently, but they make calls. They both text and text. They both, can take pictures. They both can take videos. Their mechanisms and their chips and all that are different, but they still have the same functionality. So you
Mm.
can compare them next to each other and go, well, this one, takes X number of pictures at this amount of time. This one does this. So we can have that functional discussion, and we have to start divorcing ourselves from this idea of like very specific species that is good for you, and so we need that in your microbiome versus mine, and so on. So I think we'll come to a really good place of functional analysis of the microbiome because we're getting more and more data as to what these different organisms can do and how much functional redundancy there is.
Mm. Now that kind of leads me to that next natural question, which is probiotic
Mhm.
therapy. Because I think one thing that's so interesting is there used to be just kind of like every brand had a probiotic.
Yeah.
Now we're starting to see some expansion of lines, usually targeted at maybe conditions or symptoms. But are we going to get to a place where there's, a test and then there's kind of development of a probiotic that addresses a specific function? And how far off are we from that? I know you're really well informed in
Yeah.
that industry. Like, how far off are we from a more customized approach? Because I know as a clinician, I have a few favorites for probiotics that I use,
Mhm.
but certainly I might give my number one to someone, and then someone has massive improvement in their constipation, and then patient number two has nothing, no
Yeah.
improvement. And it's really frustrating because it's a little bit of guesswork and you're like, okay, on to the next one. And it makes sense that it's not the right fit for everyone because
Yeah.
everyone's microbiomes are unique, but how could we get a more tailored approach going?
Yeah. In fact, we could probably do that today if there was enough effort and impetus put into that. We were in this weird space where probiotics and all that are driven by natural product companies, right? So dietary supplement companies. And dietary supplement companies generally don't put a lot of money and effort into research and development, right? They're kind of looking at trends and what's hot, and let's try to make something similar to this thing that's hot. And so there's a lot of tendency to move towards that market side and less tendency to do and invest in development. Pharma companies do a lot of both. They do a lot of development work, but then they also do a lot of marketing. And so where I'm starting to see that kind of therapeutic that you're referring to is moving towards the pharma side. But the pharma side, however, probiotics are not really within their scope because it's really hard to patent
Mm.
strains, right? So what they will do instead is figure out compounds that a particular strain might make that affect a functionality, and then try to isolate and patent that compound, and maybe do a synthetic version of that compound and make it a drug. So this is where we're seeing the pull, right? Because I go to a lot of these microbiome research conferences and all that. And if I ever speak at these conferences, part of my push is to companies that develop probiotics that are natural product companies, saying, hey guys, we need to put a lot more into the R&D side of this, because we can absolutely customize probiotics today based on functionality. We know enough about many strains. We can sequence the DNA of any strain. We have good high resolution sequencing. We can look at proteomics and look at what proteins every strain makes, so that if somebody needs a particular type of function, we could probably look in the library and go, aha, this is what they need. Let's develop this as a probiotic, right? So it can be done. It can be done today. The technology is there. The strain libraries are there, right? There are libraries with over 150,000 strains that you can just search the library for functionality. There's high throughput screening capabilities where you can screen
Mm.
hundreds of thousands of bacteria for particular functionality, right? And look for those specific players for very specific conditions and issues. I'm just hoping that there's going to be more market incentive for these companies to invest in that. One of the reasons why we ended up selling our business to a company called Novozymes, who's now with the acquisition of Christian Hansen, has become one of the biggest probiotic companies in the world. What I liked about them is that they had this massive strain library. I think they had about 147,000 strains, and then
Mm.
now with Christian Hansen, it's probably doubled that. And they had high throughput screening where they could screen strains for functionality. To give you an example, one of the first projects we kicked off when we got acquired was looking for probiotic bacteria that can break down BPA and
Mm.
microplastics in the body, right? And bacteria can do all kinds of things. Sure enough, in their library, they found one that can do that naturally. And so then they started the development process around that. So it's going to take companies like that. It's going to take some push for us to get there, but we could absolutely do that today.
That's awesome. All right. My last question for you, as a microbiome expert, what are like the top three things that you focus on to keep a healthy microbiome for yourself that could be relevant for other people, all of our listeners? We all want to know, how do we have a
Yeah,
great microbiome?
absolutely. So that there's so many things we can focus on, but I would say the top three. Number one is diversity in my diet. Right, I try to eat a really diverse diet to support the diversity in your microbiome. Diversity in the microbiome is by far the most important feature that we can think of in the microbiome with regards to being healthy and resilient and living a long period of time. So I try to eat as diverse of a diet as I possibly can. The second part is getting outside. There's a lot of good data on the frequency of interaction with the natural outside world, and how impactful that is on the resilience and diversity of your microbiome, right? So I'm a avid cyclist, so I'm outside a lot on trails, and I hike and love taking dogs to the walk and all that stuff, and I engage with nature as much as I can. And number three is really that management of stress. Is that finding ways that allows me to decompress, allows me to bring down the stress levels. I use Aura Ring. I love the biometrics that you can get, and this thing now has a daily stress manager, right? Or it looks at your stress patterns. So I've become very purposeful about my ability to manage stress, decompress, find joy in certain things, and then utilize that joy to improve mood and so on. And as it turns out, one of the things that is really powerful for me from a stress management perspective, and I think would be for a lot of other people, is that interaction with people with, that 16-second hugs, right? So that increasing the oxytocin, those kind of interactions, those things are really powerful for me. So that's one thing that I really focus on. Those would be the top three that I'm always conscious about. And then, of course, I take probiotics, prebiotics, and all that stuff too. But those would be the top three.
Fabulous. Well, this has been an absolutely amazing conversation. I've learned so much. I thought I knew a little bit about the microbiome, but now I realize I don't. I learned so many things today. So thanks, Kiran. This is so great.
My
And I think
pleasure.
our listeners probably loved it and took notes, and we're going to listen again. So thanks so much for joining us, and we'll chat soon.
Thank you for having me.
If you're wanting to learn and really expand your expertise in hormones, you're not going to want to miss our podcast. So make sure you tune in each and every week for our new content.